Hi, I'm Sebastian Thuault. I'm a senior editor at Nature Neuroscience, and I'm here at the Cold Spring Harbor Laboratory attending a conference entitled Brains and Behavior, Order and Disorder in the Nervous System. And sitting next to me is Adam Kepecs. So Adam is a professor here at Cold Spring Harbor, and he's interested in the neural mechanisms of cognition and decision making. And so, Adam, why don't you start to tell us a little bit about some of your research interests, some of the projects going on in your lab?
So, I'm very interested in how brains make decisions, and I'm both interested in the computational mechanisms, sort of what makes brains able to do feats that computers, even in the age of amazing AI, what makes brains do things that AI cannot do. On the other hand, I'm also very interested in the cellular mechanism at the neural level of neural circuits, sort of where's the magic in brains, what about them in terms of the biological machine that makes them able to do these things.
And so what kind of aspects of behavior do you study in the lab?
So we have a range of studies. So one of the things, for instance, what I talked about is on decision confidence. So we're interested in the neural processes behind decision confidence and try to study this in rodents so that we have the chance to sort of go down to the level of neural circuits.
And so how do you define confidence? Because obviously it's something that we used to think about when we make decisions on a daily basis, but you study that in animals, and I imagine that it needs to be properly defined so that it can be interrogated in model systems.
Exactly. So that your question sort of strikes at the heart of the problem here, which is that when we're dealing with some high-level cognitive concept, we need to be very careful and operationalize these and sort of formulate a whole way in which we can actually really make these claims. So the first set of studies that we've actually done was with humans, and we try to relate the notion of statistical confidence, which can be easily defined as the probability that an outcome, an option is going to be correct given the available evidence. So that's a formal statement, and we try to relate it to how humans respond when they're asked about their confidence. So that sort of established a framework for us that sort of laid the groundwork for the most of the actual animal work in the lab.
So what did you find in that study in humans that basically laid the ground, for further research after that?
So we found that when asked sort of in a way where we could actually define the right answer and provide in a graded way exactly, we knew what kind of evidence we provided for a person, then their confidence reports actually matched pretty much what statistics told you. So people are basically using some sort of statistical machinery, some underpinning that enables them to then report what we asked them, which is the feeling of confidence.
And so how do you then go about to translate this in the laboratory, studying more like the mechanisms in animal models? If you can tell us a little bit about the study, for instance, that you described this morning during the session.
So when we think about confidence, it's very subjective, but the reason we have confidence is because having confidence as an ability is going to make you be a better organism that's more adaptive, be able to survive. It's really a fundamental ability to essentially take the statistics of the environment, the uncertainty, summarize it, and use it for your own behavior. That's what confidence is. So any organism that's going to have this ability is going to sort of outperform organisms that do not have it. So this suggests that we don't need to sort of approach it from the level of feelings, but we can approach it from a level of a behavioral incentive where having confidence is going to make you better at it, better at a particular task than not having confidence. So we created a version of a behavioral, a simple two-choice decision behavior. So two-choice decisions is something that we and others are studying in rodents now. We can do this with olfactory stimuli or auditory stimuli, and we dial in the difficulty, and we asked.
So specifically, I mean, what do you have the animal do?
Okay. So the animal receives, say, an odor, and typically we mix it up because they're so good at this that if you give them even a pair of stereoisomers, they're going to be perfect at your answer. So perfect, that's good, but then it's not interesting because they're not uncertain about their answers. So in order to dial in the uncertainty, we mix them up in different proportions. And for the easiest ones, the animals sort of go left. Say we pair
so
one
it's
odor.
delivered, in a central port.
Yeah, there's a central port. They get an odor, and we pair one with reward on the left side. So if they get odor A, and they go to left, they're going to get a drop of water. And if they get odor B, they're going to go to the right, and then they're going to get reward. And then we mix them in different proportions, and they have to figure out what's the dominant odor. So that's the basic scenario. So that's just a classic psychophysical setup.
Right.
And then on top of it, we create an investment decision. So instead of delivering the reward immediately when it's available, we delay it according to some random schedule. So then the animal has to wait around and invest different amounts of time in order to get rewarded. And we can measure the amount of time they're willing to wait after the decision.
So that's very ingenious. I mean, that's something I really enjoyed during your presentation. So how do you come up with this idea of, to test this in the animal models? Did you try other things,
So we had a previous version of it that was a bet, a betting behavior where they had to restart. But to be honest, it's very interesting about neuroscience, we really started from a serendipitous observation about orbitofrontal cortex neurons.
Okay.
They recorded these neurons in the perceptual decision task without any confidence guided decision. And we were thinking about these issues. So sort of our minds were ready, but we didn't really know what to expect. And some of the neurons, when you looked at them, sort of, their firing patterns were strange, and they called out for an explanation. So we actually formulated a computational model to explain it, and that's when the realization came that those very strange firing patterns could be explained if these neurons were representing confidence. And then sort of we retraced our steps to figure out, okay, so how do we then ask a rat to express its confidence?
So what are the main findings that in this study? You have the behavior and you find some correlation with what you define as confidence, but you also perform some recordings.
So we're recording in this brain area called lateral orbitofrontal cortex, and that's a frontal area that has some homologs in primates as well. And this is an area that's involved in sort of valuation, inferring the values of different options in the world and outcomes. And so this area should really represent confidence when the world is ambiguous, because that's going to be your best bet to figure out what's the value of different options is. And indeed, we see different kinds of neurons functionally. And many neurons report confidence post-decision. Some of these neurons actually are correlated with the time investment behavior. So we can actually use the neural activity to predict how long an animal is going to invest in a particular decision. Then we can also turn off, if you're a psychologist and thinking about confidence, you often think about it as a metacognitive ability, because the idea is that confidence relies on a monitoring process, and this monitoring process is in that sense meta, but it would also imply that the confidence representation ought to be generalized and not specific to any sort of sensory stimulation. So one of the very recent studies we've done is record these neurons in a dual task where we have auditory and olfactory decisions, and can show that the same neurons represent confidence about both of these decision types. And then we can also inactivate this brain area. That's one of the advantages of working with rats, that we can do a lot of invasive experiments as well, where we can really interrogate the neural mechanisms of what's going on, and we can turn off this region.
So you're talking about the OFC here.
The OFC.
Yeah. So you found something interesting actually when you turned off that region in terms of the behavior.
Yeah. So exactly. So by turning off the area, what we found is that the decisions themselves weren't impacted at all, which makes sense, that these are perceptual decisions, but the time investment became random. It's not that animals spent more or less time waiting for reward, but they were no longer predictive of accuracy. So they were essentially not as optimal in investing time.
So it's as if they couldn't, have any confidence
Exactly.
in what they saw. So, and in terms of the, I imagine you saw different types of electrophysical behavior when you recorded in the UFC. You saw those neurons that attract confidence. Did you find other kinds of responses there?
Right. So there's lots of other kinds of responses. That's one of the challenges that we face in cortical physiology, especially in frontal cortex. So one thing we can do is manipulate different contingencies. We can say change the reward size, and then can ask the questions, do these neurons vary with reward size as well? So one of the observations we made is that there are neurons that specialize in confidence, and they don't care about reward. There are neurons that specialize about reward, and they don't care about confidence, the reward size. And there are neurons that integrate the two and always do so sort of the right way, so that they represent either high value or low value. So these are the functional classes. They also have different time courses often. So one of the things we're thinking about now is how to connect these differences in the functional types to the neuronal cell types. And I think that's one of the critical challenges that we face in systems neuroscience today, is to relate sort of the incredible diversity, but also exquisite precision of different cell types in cortex to behavior.
Right. And so, so that you're talking basically about future work. And I know you're very much interested in cell types. So what would be the next step? I mean, are you willing to discuss some of this today?
So it's very challenging. So now, we've gone through an incredible revolution in neuroscience, and it's so exciting to be able to do experiments that we couldn't have imagined even a few years ago, really. So we have a number of different ways in which we're trying to get access to the cell types. So one is we use this method called optogenetic tagging. So we put a channel rhodopsin, a light activatable channel, into specific projection types, specific neurons that go to certain target areas and not others. We know that some of these neuron types sort of sort themselves out. So one type sort of goes to one brain region. And along with our electrodes, we put a tiny optical fiber in, and we're trying to activate them, but not to just change behavior, but to use it as sort of a beacon to figure out what cell type it is. And so this way we're trying to, for instance, figure out what are the different cell types.
So you have an interest in interneurons?
That's right.
So do you already have a few pieces of data that sort of illustrate what could be the function of these guys in this region? Or?
Yeah, so we're just starting in OFC with inhibitory neurons, partly because all of this work we do in rats, and in rats it's a little bit harder to target genetically some of these cell types. But we've done a lot of work in mice, and some of those studies are not about confidence, but they're related to frontal cortical questions, and fundamentally trying to address the things that are sort of accessible in mice, which is these different cell types. So we know that some of the major cell types, the cardinal inhibitory neuron types, have very different behavioral functions. So PV, somatostatin, and VIP types of neurons, which probably also have their own subtypes, they do respond in very unique ways. So we're trying to figure out the principles of that.
Because I guess, one of the interesting question now, you see this diversity of cellular behavior in this region. I guess one of the questions is how these neurons interact and what kind of information is integrated, and perhaps what kind of circuit motifs are important to process the information. So how do you think this can be best addressed, as a sort of a next step? Could you have a theoretical background? Is this something that could be used to generate some hypothesis, or?
I think there's a lot of hypotheses. I think what's changed the world is sort of all these new technologies that really allow us to interrogate things. So until quite recently, when you were interested in cell types, you could only use reduced preparations. You could take them out of the brain. You could look at anatomy. You could fill it up in vitro. And I think what really changed is that now, you can do all of that at a sort of a much greater throughput, but in addition, what you can do is interrogate these in a behaving animal. And that sort of gives you access to a whole new kinds of new dimensions in terms of what their functions could be. And I think the issue that it raises really is that there's a debate that's still ongoing of what's the correct way to categorize cell types. And with all these sequencing technologies, RNA-seq, where you can really have incredible information about single cells and these anatomical mapping projects, we have a lot of hypotheses about how to categorize cell types. But I would argue that we also need to include functional criteria, because ultimately the right categorization should include, in some sense, these neurons should behave similar ways.
Yep. No, that's I agree. That's very interesting. And there are lots of questions arising from these, single cell RNA sequencing studies. So I have a couple of questions. I mean, I know you have a very strong interest in neuromodulation also, and decision making is going to really depend on context, on external variables that influence perhaps brain states and things like this. Is this also something you're exploring?
Yes. So neuromodulation is something I'm very excited about. So neuromodulators basically sit at the bottom of the brain and project across many brain regions, and that makes them sort of unique in their ability to reconfigure circuits. And there's been a lot of computational hypotheses put forward about these. So those are, strongly influence our work. And again, there's a lot of new tools where we finally are able to interrogate these systems. So that's something that's very exciting for us. And in addition, some of our work is actually connecting with the inhibitory neurons because we think they might actually exert some of their postsynaptic effects through specific subtypes of neurons.
Okay. And something that struck me this morning is that your presentation was in the psychiatric neuroscience session. So I wanted to ask you about how you see this kind of work being important in the context, in the clinical realm. How can it be important for translational neuroscience? What are your thoughts on this? And if this is something that you're interested in developing in your lab?
Yeah, I'm very, very interested in biological psychiatry, and we started a number of different studies along this line. The basic challenge for psychiatry is that the symptoms are very subjective, and we know that they derive from something going wrong in the brain. And it's very difficult to address in a rigorous scientific way. And I think neuroscience is really faced with the challenge of how do we connect our work to something that's useful for psychiatrists. And I think some of our work in cognition is useful because in order for us to establish the notion that operationally rats can express their confidence, provides you with a quantitative tool that we can use for humans as well. And we can create metrics that can individually measure sort of distortions in confidence in patients. So that's one line of research we're doing. And on the flip side of it, in animals, because we can do the same things, we're really interested in understanding how some of these dimensions that are useful for psychiatry, what are their underlying circuits.
Well, that's great. That sounds very exciting, some future discovery. I'd like to end our conversation with what I call, or what we call a micro-interview. I actually borrowed that format from some of my colleagues at Nature Microbiology. And basically, I'm going to ask you a series of simple questions. You have to make a choice. You have, those answers can only be 1 or 2 word answers. And you have 1 minute to answer as many questions as possible.
No confidence reports, no?
So are you ready? I'm going to set the timer. Okay. Rate code or temporal code?
Rate code.
Parietal or prefrontal?
Prefrontal.
Top down or bottom up?
Top down.
Olfactory or auditory?
Auditory.
Noise correlations or signal correlations?
Noise correlations.
Low dimensional or high dimensional?
High dimensional.
Somatostatin, parvalbumin or VIP?
VIP for sure.
Rats or mice?
Pass.
Electrode or microscope?
Electrode.
Empirical or theoretical?
That's not fair.
You can say both. You could say both.
Both.
Optogenetics or chemogenetics?
Both.
Silencing or activating?
Silencing.
Head fixed or fully moving?
Both.
Big lab or small labs?
Medium.
East Coast or West Coast? Oh, we're done.
East Coast.
Well, that was great. Thanks a lot, Adam. It was a pleasure to have this discussion today with you, and I look forward to all the new development in your lab.
Thank you. It was great talking.